Very good morning to all of you. So let me start with an introduction of myself, which I avoided in the elective session because I knew that uh, there is going to be a bigger class than this in the very next lecture. So I avoided introducing myself in detail there, but I think this is the right time for me to introduce myself to you guys. So my name is Sachin Rajgopalan, and I have completed my bachelor's and master's in the subject of microbiology from SIS College Science. And post which I was working with an industry called Advanced Enzyme Technology for around 6 months because my aim was to get into teaching. So I was waiting for my entrances to be cleared. So I cleared my net and set in life sciences, and I got into Department of Microbiology, Ramnarayan Rhea Autonomous College in 2016. So since past 5 years, this is my 6th year in Rhea College that I have been teaching bachelor's and master's students of microbiology and vocational course of pharma analytical sciences. So these are the three courses that I basically deal with at Rhea College. And other than that, because of my core interest in psychology, I am pursuing my master's in psychology at this point of time because of my liking for neurology and neuropsychology. So that has motivated me to do that part also in arts. So that's how I've been handling both the things at the same time. As far as my research interests are concerned, currently I'm pursuing my PhD in antibiotic resistance and biofilm management at Rhea College itself in association with Dr. Rajesh Patkar from IIT Bombay. And other than that, I have guided around 30 plus MSc dissertations, wherein I love to work in the area of quorum sensing, understanding the environmental issues and trying to find out solutions to it with microorganisms. And one such initiative that we had as a group project with our TYBSc students in 2018 was wherein we genetically engineered Escherichia coli to degrade palm stains, and we represented the country in genetic engineering based projects in Boston in a competition called International Genetically Engineered Machines, which is abbreviated as IGEM, basically started by MIT Boston, Massachusetts Institute of Technology. And fortunately, our team won the gold medal and also a Best Human Integrated Practices Award amongst 385 global universities. We won that award. So my research basically revolves around, my research interest basically revolves around dealing with ecological issues and trying to find out solutions for those. Though from my MSc days, I was very, very interested in cell biology, but the opportunities of actually working with the cell lines has been not that much, because of which I couldn't explore that area much, and I diverted myself into understanding the ecological issues. But very recently, we have started working on drug resistance because MDR has become a global issue. So we are working on MDR as a basic problem with collaborations with NCCS Pune, and we also have some collaborations with Systems Biology Institute in Seattle, USA. So there are some projects working in that area also. So these are my brief, prospects in terms of research. But yes, my soul, my heart lies amongst the students. It is teaching, and that's the reason why probably I'm here since the last 5 to 6 years now. So Somaya has been very, very close to heart because I've been visiting the institute, especially the Department of Microbiology, for a lot of examinations. Very close friends and colleagues are there in the department there, even in the Department of Biotech, wherein I have got opportunity to work with people, with faculty members there on different instances. And it is really great to be a part of the system and I hope that my association helps you out in understanding the subject even 0.001% more. I will consider it to be my success. So I will be dealing with one of your unit, which is on genome organization and DNA replication. Let me tell you that this particular unit is quite fundamental in genetics, in understanding of eukaryotic and prokaryotic genetics. And it's going to be helpful to you not just now, but even during your master's course, even during when you give entrances for admissions into good institution for masters, or even for your admissions for PhD, your interviews that you attend, because this is core fundamentals of genetics, right? So we will be dealing with that, and we will be going step by step and not rushing into the concepts. I will tell you what my ideology is going to be in dealing with this, and we can adapt as per your requirements. That is completely fine. Okay, so first and foremost, I have told the students of the GE also that I will be passing on all materials to you guys. I will make a wiki link. I will show you what a wiki link is, because I've already created one for the genome organization unit. So I'll be sharing that link with you guys. Let me put that in the chat box first. I will also send an email with the link for the same. And I will also cast my screen and show you what a wiki link means. Okay, so those who are on laptops or desktops and can open a different tab can open this link and visualize. For the others, I will cast the screen. Okay, so just give me a minute to cast the screen. Can any one of you tell me if the screen is visible to you guys? If yes, just let me know.
Yes, sir.
Yes, sir.
Great, thank you so much. So this is how a wiki link would look like. You will see that there are different sections made. Okay, right now these sections are empty. Only this section is having something, that's the reference book. So I will show you what's that. So whatever sessions we will conduct, all that will be recorded. I will post all the recordings with what we discussed in that section as the title of that, and I'll post it over here under the recording section so that you can refer to it to revise the concepts. Not today, but I have this habit that after every lecture, I normally give a link to students, which is a Google form link, wherein there will be few questions about what we discussed today. They will be like MCQ based questions, or you may have to type a one-word answer. And that will be to check how much you have been able to assimilate in the session. So we won't do that today, but we will do it from the next time. So all those Google form links will come in the assessment section. It will not be marked anywhere. It is for you to understand and for me to understand how much you have been able to assimilate in the process. All PPTs that I will use, though today I have no PPTs with me, we will be discussing in general about what the concepts we are going to learn. All those PPTs will be put up here. There will be some videos that I show you. There will be some extra reading materials for you to get more information. All that will come in the extra reading section. Okay, so you can see right now that I have made a Google Drive folder for reference books, and I've uploaded all the reference books out there. So when you click on that, it will take you to a Google Drive. I'm sharing the Google Drive screen with you guys. You will be able to see now all the books that have been prescribed that have been suggested in your syllabi, all that have been put up over here. So you don't have to worry. I'm going to try my level best to get everything for you at one place, so that your job is to just sit and study. Okay, so this wiki link is going to help us do that, right? And at any point of time, please understand that all are learners here. Okay, you are a learner, I am a learner too. It doesn't mean that I know everything if I am teaching something to you guys. Okay, even I am a learner. So feel free to ask questions. Okay, I will reiterate this, that it doesn't mean that I will be able to answer all those questions right away. But if I feel that there's something that I cannot answer, I'll surely get back to you once I read about it. But that shouldn't stop you from asking questions. When you ask questions, that's when curiosity gets an impetus, and you start thinking about it. So please speak. You are free to unmute and speak at any point of time in my class. I'm not going to tell you that why did you speak in between when I'm explaining something. That's not going to happen. Okay, you can unmute and speak at any point of time. Even if there is background disturbance, please do not hesitate to unmute yourself. We all are adapting to this mode. We all know that what are the repercussions of this platform, but that shouldn't stop you from speaking. So feel free to unmute at any point of time, shoot your questions, I'll be more than happy to answer them. Okay? If you still feel that there are some issues and you don't want to unmute yourself, then the chat box is always there. Feel free to put your questions, put your comments in the chat box. Okay? Any kind of feedback is also welcome. You can tell me that, sir, ye hum aise karte hain kya? I'm open to it. Okay, I'm completely open to it. It's, as I said, it's a learning medium for all of us. Okay, learning never stops in life. So I will be more than happy to hear from you guys, and we can adapt in the due course of time as we discuss the topics. Okay, so please do not hesitate to ask questions. Okay, other than that, please take pointers as we discuss concepts, because genetics is vast. And I always feel that, when you are absorbing things for the first time and you note things down, it remains there. And when you read it, it becomes very easy for you to remember. And also very easy for you to refer to the book, that what did we discuss and what you are expected to read from the book. Because books are always vast. They have lots of things given there. It doesn't mean that everything is going to be a part of your curriculum. Not at all. So you should have things written as pointers. I don't say that gada majuri karo, sab kuch word to word likho. No, pointers mein likho. Important points note karo, ताकि आपको easy हो जाए relate करने के लिए कि हमने क्या discuss किया था, कितने limit तक discuss किया था, और कितना आपको read करना होगा. Okay, reading आपके ऊपर है, आपको जितना read करना है आप read कर सकते हो. But from the limits of the boundaries of the curriculum, you should know till where you are expected to go, right? Okay, so I really hope that this will be very interactive session for both of us, and we will be able to take out something of these 15 sessions that we will conduct for this particular unit. Okay, another thing, I am very particular about time. I prefer to start on time, stop on time, so that lectures don't get extended, and we are not, we should always value time. So time is precious. We will stick to what time slots have been given to us. Okay, great. So I think we are all set to begin with our discussion. So to begin with, you all know what genetics is about, right? It is basically the study of the genome and the constituents of the genome, which takes us to the understanding of how, or rather what guidance is given to the cell to perform specific set of functions. I always say, when people say the organization of organisms, especially humans, hum wo school mein padhte hain, that an organism is made up of organ systems. Organ systems are made up of organs. Organs are made up of tissues, and tissue is made up of cell. And we have that Ghisa Pitta line, which you see in your textbook every time. Cell is the basic structural and functional unit of life, right? But I refute this. Refute this means I do not defy that this is not a fact. But I would rather prefer to say that cell is a basic structural and functional unit, which is guided by a unit below it, which is the nucleic acid. Because without that nucleic acid, no organism can survive, because the guidance is that nucleic acid. So until and unless I get that guidance, I don't know what to do. I don't know how to shape myself. I don't know how to develop myself. I don't know what function am I expected to carry out. So if I say that my entire system is functioning on the basis of that one nucleic acid molecule, and I know that I have trillions of cells in my system and every cell is doing something different and unique, the confusion arises is that small molecule, which is same in all the cells of my system, is still able to carry out a plethora of different kinds of functions. It's able to guide the cell to specifically act in a specific way in a specific circumstance, such as small molecule. How does it do that? Because the cells of my body are not visible to my naked eye. I am talking about a molecule which is much smaller than that, which fits into a small compartment of the cell, which I refer to as nucleus. So isn't that amazing that I have something so tiny which is guiding me, my system, to carry out the entire work? And that too, it's the same molecule in every cell, but the function that it makes the different cells perform is different. That's where we learn to understand how beautifully our DNA is packaged and how beautifully my DNA is regulated. So here we come to understand the concept of genetics and the concept of gene regulation. And for that, understanding the organization of my genetic material is very important. Understanding how my genetic material is packaged to become so compact a structure becomes important. So that's where understanding the genome size, how it is compacted, how it is organized comes into the picture. And more importantly, in this unit, our focus will be to understand the basic differences between a prokaryotic genome and a eukaryotic genome. What are its constituent genes? How is it packaged? What are the basic differences that you will see in both the genome? How big or small these genomes are from each other, and still how complicated they are. We will be seeing this. Let me tell you that when you talk about yeast, Saccharomyces cerevisiae, best example, or when you talk about something like Caenorhabditis elegans, worm, chota sa worm, or when you talk about mus musculus, that's the mice, or you talk about monkeys, or you talk about humans, compare their genomes. The data that comes out to you is mind-blowing. Because if I compare the yeast genome and the human genome, I find that there are around 60 to 70% similar genes. Such a tiny microscopic organism, which is not even visible to my naked eye, is sharing almost a little more than half of the genome that I have, or that I am made up of. The small worm, so tiny, it is sharing about 70 to 75% of the genome that I have. Or the rat, look at their size and look at our size, they are sharing about 98% of the genome that we have. Or talk about monkeys, our ancestors. They share almost 98 to 99% of the genome that we have. And that's the basic reason why you end up using rats and monkeys to test anything before you test it on humans. Though there are obviously a lot of animal ethics involved there, but that's the reason why, testing on these animals began. So what does this tell us? This is a beautiful proof of evolution process. This is telling you that we all organisms have evolved from each other. This is telling you how complicated organisms like us have evolved from microscopic, unicellular, prokaryotic organisms, which are so tiny, and still they are carrying out complicated functions out there. They don't have a brain like you, but they can still make your life difficult. I think we are living that situation right now, wherein a small, tiny virus has screwed lives, has screwed the world. Please understand the potential of the subject that you guys are studying. It is a phenomenal area. Whenever students come and ask me that, microbiology or biotechnology ko kya scope hai? It's a very ridiculous question, believe me. मेरा जवाब एक ही होता है, स्कोप किसी भी सब्जेक्ट में नहीं होता, तुम अगर 100 साल पहले किसी को बोलते कि हाँ तुम अमेरिका में बैठो, हम फोन पे से एक दूसरे को देख के बात कर पाएंगे, तो लोग तुम पे हंसते, लोग बोलते ये तो पॉसिबल ही नहीं है, इसमें कोई स्कोप ही नहीं है। बट इट्स अ फैक्ट टुडे, फेस टाइम इज अ फैक्ट टुडे। सो स्कोप होता नहीं है, स्कोप बनाना पड़ता है। Okay, very difficult to accept this fact, but this is true. We all have to struggle our ways out to generate scope in the area that we like. This is the time to prove that our subject has so much of potential. We can do so much out there. So learning these fundamentals should pave your way towards coming out with new ideas. So think, let your thought process evolve, just like we have evolved. Okay. Now in this unit, we will be dealing with the eukaryotic genome, wherein we take example of human genome. We try to understand the organization of human genome. We try to understand the number of genes present, the types of genes present, how do we understand their functions, okay, how they are categorized on the basis of their functions. This we will be basically focusing on. And then we will moving ahead to understand the prokaryotic genome, which is, a little different from what eukaryotic genome is in terms of the constituents, in terms of the organization, in terms of the genes that they have, the functions the genes perform. They are quite different. And it also helps you then understand how the basic processes of replication, transcription, translation might have slight differences. The process may remain the same. The crux of the process may remain the same, but there are going to be small differences always involved in the process. We will also try to understand a concept which has been very popular. You've studied it in 12th standard, which we call as operons, wherein you try to understand that when you have a set of genes functioning together, how does it bring about a change, which has been very popularly studied in prokaryotes. The question is that why don't we have them in eukaryotes? Or rather, I would ask the question, do we have it in eukaryotes? Maybe yes. So you should start reading about that before we actually come to that concept of operon. Because that's very interesting to see that how people are searching that do we have concepts similar to operon in at least lower eukaryotes, if not higher eukaryotes. Like yeast, fungi are lower eukaryotic organisms. So can we see a similar concept like operon there? Big question. And we should have a look at it. Right? Then we will move to a very important concept, which is the DNA replication process. How it was studied, how it happens. There are some experiments that we will study, wherein we understand that how copies of DNA is going to be made. And why is it so important? What happens if something goes wrong there? Now, I want you to tell me that why is it so important to learn genetics? Can you tell me? What's the significance of learning this subject? Anyone? Unmute yourself, write in the chat box, completely fine. Why is it so important to learn genetics? And then I will give you my ideology of why it is significant to learn genetics. Anyone? Come on, guys, you have to interact. Yes. You want me to name you guys? I don't want to do that. So please unmute and speak by yourself. Diagnosis of genetic disorders. Okay. Anyone else? Anyone else? That's it. हर कुछ चीज हम जो करते हैं वो बीमारी पढ़ने के लिए करते हैं. Very good. Helps in understanding the genetic makeup. Very good. Anything else? Excellent. Phylogenetic analysis. You want to study how, the genome of different organisms are related. So you establish a relationship between the organisms that helps you generate the evolutionary chart, the evolutionary tree, right? Okay, very good. Then, anything else? Come on, come on, come on. Okay. So first and foremost, the aim of understanding the genome of any organism is to understand how that organism functions, what are the different genes they possess, why they may be present, because that helps you understand the physiological processes in that organism in a much better way. Okay? And once you study the genome, it gives you an idea that what would go wrong in case of an abnormality. So you automatically end up studying genetic disorders associated to the different genes that might be present in that genome. Very important, right? Because एक टाइम था जब हमको पता ही नहीं चलता था कि एक जेनेटिक डिसऑर्डर है तो ये फर्स्ट ऑफ ऑल जेनेटिक है, यही नहीं समझता. With the advent of the study of genetics, with the advent of Human Genome Project, the things have become much clearer that, okay, these are the genes present. If a person is suffering from this kind of a genetic disorder, this is the gene at fault. This might have gone wrong. At least this knowledge is available. That helps you in a way to design different therapeutic modules. I don't say that all genetic disorders today are curable, but at least you know what can go wrong and what can I do to at least reduce the symptoms that the person is suffering from. Or if I'm trying to design a gene therapy, then what should I exactly do in this particular scenario? Let me tell you, students, that this is the era of personalization. Everything, every damn thing is being personalized. Okay? So you are personalizing your costumes, you are personalizing your accessories, you are personalizing space, you are also personalizing your system. Let me tell you that today, with the advent of genetic engineering, there has been so many ethical concerns raised to it, because there's a concept called designer babies. I don't know how many of you have heard about it. So with the advent of gene editing tools, especially with CRISPR-Cas, there's a scare. Because tomorrow a couple can walk into a fertility clinic and they can say, they can place an order. These are our gametes. These are the characteristics that we are looking for. We want the baby to have this height. We want the baby to have this kind of IQ. We want the baby to have this eye color. And the fertility clinic has to work on it. Now, that's a big ethical issue. Because you are no one to decide for an individual who is going to be born. Maybe that individual doesn't even want to be an athlete. Why are you deciding how tall he or she should be? And moreover, if people start working on IQ using these techniques, फिर तो बर्बाद थे. अमीर अमीर होते जाएंगे, गरीब गरीब ही रह जाएंगे, because ये expensive होगा. So this technology should be used for the benefit where it is needed, like working on disorders, and not on, doing these things which are unethical. Secondly, we need to understand that understanding your genetic makeup is going to give you an idea of how your body is going to respond to certain circumstances. As very correctly in the chat box, Pranav has written that several disorders may not be directly related to your genome, but how your genetic makeup is, that predisposes you, that increases your chance of getting a particular condition. So that is again significant. To a great extent, people are understanding or trying to understand how genetics can influence your behavioral aspects as well. How genetics can influence your thought process as well. Wherein you're trying to, amalgamate two different fields of neurology and genetics. To a great extent, let me tell you, the future of medicine is going to look very different. there is going to be an advent of personalized or tailor-made drugs, which means that there is going to be a time, the way you have Aadhaar card right now, you will run with a chip. You will have a chip with you, a very tiny chip, which will become your medical identity. What is that chip going to have? It's going to have your entire genome sequence. There's going to be a time when every individual will have to get their genome sequence. Maybe not in 10 years down the lane, but in the next 20 years, this is going to be the fact. Everyone will have their genome sequence. Now I'll tell you why. I'll give you an example. I'm going to take names from the class here to make it relatable. Let us say that Prerna is a physician and I, Nidhi, Sonalika and Pranav are patients. We all are having the same condition, same symptoms. We go to Dr. Prerna, we tell her our symptoms. Prerna sees that we all are having similar kind of symptoms, similar kind of condition, and she prescribes us the same drug, the same dosage. And she tells, complete this course, get back to me and tell me how it is. Now Nidhi, after consuming the very first dose, is feeling very good. She is in complete awe of Prerna's ability of treating, because she has got good relief. Sonalika, after 3 doses, feels a little better, but she struggled till that time. I am taking 4 days to feel a little better after the dosage, while Pranav doesn't feel good at all after completing the course as well. The condition still persists. So we all have gone back. Nidhi is giving a very good response saying that I felt very good after the very first dose. Sonalika says that it took me 3 doses, but I was completely fine. While I tell her that I took 3 to 4 days to feel better. While Pranav is pissed off that the course didn't help him in any way. Now, ये प्रेरणा को as a doctor ये idea देगा कि ये 4 अलग system है. भले इन चारों का problem एक है, but इनको same kind of drug, same kind of dosage देके चलने वाला नहीं. Now why? There are several reasons to this. First, your physique matters. What's your height? What's your weight? That tells you that how much dosage or concentration of drug will you require in your body. Now let us say that Prerna gave us a very good amount of dosage which suits all. So physique was not the problem. But the problem is in the genetic makeup. I can't even call it a problem. It is a variation in the genetic makeup. How you respond to a particular drug, that is decided by your genetic makeup. For some, let me tell you that, any kind of antibiotics or drugs that you consume, you have to understand that that is foreign for your system. It's completely a foreign material for your system. So after some point of time, the liver is going to recognize, okay, this is a foreign material in the blood. I need to excrete it out. That's how you start excreting out drugs in the urine, or their metabolized forms in the urine. Now, for Prerna, she got enough dosage in her system. It acted well on her system, because she was able to retain the drug for a good amount of time in her body. She felt good. For Nidhi, right? For Sonalika, she had to take 3 dosages, which means that to attain that threshold concentration in her system, she had to take 3 dosages, because probably the system had started eliminating the drug. For me, it took even longer, because my system is actively eliminating the drug from the body, but it took some time for it to accumulate a good amount in my body. But for Pranav, it's not at all getting accumulated because his system is eliminating the drug at a very fast rate from the body. So even before the second dose is going in, the first dose is out. See, this is the reason why when you go to the doctor tells you that 3 dosages है, एक सुबह खाओ, एक दोपहर को खाओ, एक रात को खाओ. क्योंकि उसको पता है कि जो तुम सुबह ठूसोगे वो दोपहर तक तुम्हारी बॉडी से बाहर जा चुका होगा, जो दोपहर को कंज्यूम करोगे वो रात तक बाहर जा चुका. And maybe for some individuals, let us take another person here, let us say Kushi is consuming the same drug. She, after consuming the drugs, after 2 or 3 dosages, she has started exhibiting completely different symptoms now. मतलब दवाई खाके उसको कुछ हो रहा है. Now that could be because of 2 reasons. One, she could be allergic to the constituents of the drug, but let us say she's not allergic. Then the second cause for this is going to be that her body is not eliminating the drug at all. Very slow in doing the elimination. If the drug accumulates too much in concentration, leads to toxicity. Why is this taking place? We all are of different genetic makeups. Let me tell you that if we compare our genome at this point of time, 99.9% of our genome is going to be the same amongst we humans. What is different is 0.1%, and that 0.1% is responsible for the huge amount of variations that we have. So can you imagine what is that 0.1% which gives you the identity that you have? A different face, a different body architecture, a different kind of behavioral aspects, a different kind of physiology, that 0.1%. That makes the process interesting. We are here to study that 0.1%. So there is going to be a time when your entire genome will be sequenced. You'll go to a doctor, the doctor will say that tell me your accession number or give me the chip. The moment he or she loads that chip or puts the accession number, your entire details will come on the screen. Your entire genome sequence will come on the screen. All your medical history will come on the screen. And then the doctor, according to your genome, will connect it to a database, and the database will generate what are the drugs which can act on this individual, which drugs will not work. The drugs will become tailor-made, personalized drugs. ना इसका सबसे बड़ा फटका किसको बैठेगा? फार्मा इंडस्ट्री. क्योंकि हमारे देश में स्पेशली जहां पर इतना बड़ा पॉपुलेशन साइज है, drugs are manufactured in bulk. We are bulk manufacturers of drugs. Tons and tons of tablets are produced of the same thing. Isn't it? But when I start giving personalized drugs, the manufacturing process will get affected. But this will eventually happen. This has to happen. And this is where you have the area of pharmacogenomics, pharmacogenetics that comes into the picture. Pharmacogenomics is of much interest to pharma industries, wherein they study that human genome, how will it respond to drug A? So in general, they're studying in general, what's the response of a human being to this drug on the basis of the human genome makeup. But then we have to address for that 0.1%, which is different in every human. So there comes pharmacogenetics, wherein we discuss how an individual human being will respond to that drug, where it's not a general profile, it's a person-based response. To see how significant the study of genetics becomes. And for this, your understanding of genome organization, the gene constituents, is very, very important. And especially when you are a person who is interested in understanding disease physiologies, disorder physiologies, you love to understand what is oncology or cancer biology. This is the unit for you guys. So we will be focusing on the natural process of replication, what happens when it goes corrupt. Both the aspects we will try to analyze. So our flow is going to be wherein we start first with the prokaryotic genome, because I feel that's the correct order of doing it. You discuss the prokaryotic genome first, understand the nucleoid organization, try to understand the constituents of the prokaryotic genome, the operon concept, and then gradually we move towards eukaryotic genome, where we take humans as an example and human genome as an example, and then move towards DNA replication. Because prokaryotes have ultimately evolved to eukaryotes. And when you see the eukaryotic organelles like mitochondria and chloroplast, you will see that the genome that mitochondria and chloroplast have is nothing but the prokaryotic genome, because we know the endosymbiotic theory. We know how prokaryotes have got converted into the eukaryotic cell organelles. So we have to understand the prokaryotic system, the prokaryotic genome first before we move to the eukaryotic. So that's the reason why I'll first start with the prokaryotic genome, then go to eukaryotic genome. Okay? And then finally, we will dedicate our time to DNA replication. Now, when we study prokaryotic genome, we need to understand that it's not just the nucleoid, it's not just the chromosomal component which is present. There may be extra chromosomal components also. And there is where we come across a very beautiful, a very challenging concept for the human mankind, where we discuss about plasmids. Because these plasmids, if they have made the human life much better by commercialization, they have also made it worse by coming up with multiple drug resistance. So we will be discussing about plasmids as well in brief, because that's a part of the entire, what you call as the genome or genetic constitution of a prokaryotic organism. So we'll be also focusing on the plasmids. Okay, so do you have any questions that about the concepts that we have discussed till this point? Please let me know if you have any specific questions to ask. Anyone, you can unmute, you can type, it's all up to you. Anything, any queries? Are you understanding what we have discussed till this point? Can I at least see a yes in the chat box? It's very important for me to know that if you are able to relate to what I'm speaking over here. Great. Okay. Amazing. Fine. So, can you tell me what is the difference, basic differences between a prokaryotic cell and a eukaryotic cell? Come on.
Sir, compartmentalization.
Excellent. Compartmentalization. What else? What else? Nothing else. What's the major criteria of differentiation? What's the major criteria of differentiation? अरे तुमने प्रोकैरियोट यूकैरियोट का डेफिनेशन पढ़ा है ना? प्रोकैरियोन, यूकैरियोन. Anyone? Come on. I know you all know it. You want me to name you guys? I can do that. So yes. Aditya, can you tell me what is the meaning of a prokaryotic cell?
Sir, the basic difference, the prokaryotic cell means not developed, means the whole cell, and the difference is that the eukaryotic cell has a membrane-bound nucleus, while the prokaryotic cell do not have membrane-bound nucleus.
Okay. So it's all about वेल डिफाइंड न्यूक्लियस, राइट? सो कंपार्टमेंटलाइजेशन जब हम बोलते हैं तो उसमें ऑर्गेनल्स भी आता है, उसमें न्यूक्लियस भी आता है. So when you talk about prokaryotic cells, we say that doesn't have a well-defined nucleus, a very rigid way of defining it though. And eukaryotic cells, we say that their genetic material is present inside a separate compartment which we refer to as a nucleus, right? So in prokaryotic cells, you have your genetic material which is in the cytoplasm itself. It is in the form of a very condensed structure, not as condensed as the eukaryotic genome is, but a condensed form which is called as nucleoid. So is the nucleoid free floating in the cytoplasm? Can someone tell me about that? Is nucleoid free floating in the cytoplasm of prokaryotic cell? Yes. Have you understood the question? उसके लिए तो yes or no बोल सकते हो. Have you understood the question? Yes. Only one person in the class. Others have not understood it. See, DNA in case of prokaryotic cells is in the form of a nucleoid. So is that nucleoid, there is no compartment, so it's not being restricted. Is that nucleoid floating in the cytoplasm everywhere in the prokaryotic cell? That's the question. Or is it localized in some way? You have studied this in 11th standard. I'm sure about it. Does anyone remember there was a chapter in 11th standard called as Organization of Cell? Biology. Yes? Do you remember? For CBSE students, it was cell, the unit of life. Do you remember there you used to study the prokaryotic cell structure? You used to say that plasma membrane has an invagination, it folds towards the internal side. What was that invagination called? Anyone? No? Do you remember this term? No? Not at all? Arey baap re. Difficult hai. Okay. So, when you used to draw the structure of a prokaryotic cell, you used to say that there is a cell wall and there is a plasma membrane. So when you used to draw the plasma membrane, you used to show an internal invagination. And you used to say that irregularly folded DNA is going to be attached to this invagination, which you used to call as mesosome. This mesosome is responsible for holding the DNA at one place. It is also responsible for initiating DNA replication. It is responsible for ensuring that the copies of the DNA is distributed into the two daughter cells in case of prokaryotic cells. ये सब हमने 11th standard में पढ़ा था. So you know this is what we have to understand. It's okay to not know things, but it is very important that when we learn things, we try to at least keep in touch with the concepts that we have learned. Or else it's going to be difficult for us at a later point of time, because फिर पीछे मुड़ के देखोगे तो it looks like an ocean. तो तकलीफ होगा, क्योंकि अभी तुम बीएससी के बाद तुमको अगर एमएससी करना है, अच्छे इंस्टीट्यूट्स में एमएससी करना है, तो एंट्रेंसेस देने पड़ेंगे. एंट्रेंसेस सिर्फ देके नहीं होता, एंट्रेंसेस क्लियर करने के बाद इंटरव्यू देना पड़ता है. तो वहां पर अगर आप चुप रहोगे, कुछ नहीं बोलोगे, तो फिर सिलेक्शन नहीं हो पाएगा. बहुत ही कॉम्पिटिटिव एरिया है. तो गलत होना इज फाइन. देयर इज नथिंग रॉन्ग टू गो रॉन्ग, टू मेक मिस्टेक्स. वी आर ऑल ह्यूमंस. सो वो बोल के अपने आप को सप्रेस करके रखना, बात नहीं करना, इज नॉट अ गुड ऑप्शन. So I'm really hoping that the next time I see you guys, I will be able to see you guys interacting. Yes? Yes or no?
Yes, sir.
So please start speaking up. It's very, very important. I'm sure that you all know concepts. It's just the matter of confidence. Get that confidence in you. It is okay if you go wrong. We are here to correct you. इधर गलती करोगे चलेगा, उधर जाके गलती करने से तो बेटर है ना. Okay, so at this point we will stop for today and we will meet next Thursday and we will start with prokaryotic genome organization first. ठीक है? If there are any questions, please feel free to ask. Keep checking the wiki. I will also share the wiki link by email so that you can constantly keep a check. You can bookmark the wiki link so that it's easy for you to access anytime. Okay, thank you so much and see you all in the next week. Bye bye. Take care.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.